continues to advance the natural product research and pharmaceutical sciences, bridging traditional knowledge with modern innovation. So I big round of applause to our chairperson, Professor Sanjeev. So I can just tell you, so in this session, we have only one speaker, and our speaker is Professor Avinash Bajaj from RCB. So I would just ask the chairperson to introduce the speaker.
Assalamu alaikum and very good afternoon to one and all. It's my pleasure to introduce the speaker of this session, Professor Avinash Bajaj. Dr. Bajaj is a distinguished researcher specializing in nanomedicine, cancer immunotherapy, and biomaterial for drug delivery. He was a Ramanujan Fellow, and just to apprise the August gathering that Ramanujan Fellowship is a very prestigious fellowship of the country. His research focuses on the developing lipid nanoparticle for tumor targeting hydrogel-based drug delivery system and exploring sphingolipid metabolism in cancer. With multiple high-impact publications, Dr. Bajaj continues to drive innovation in cancer treatment, antimicrobial therapy, and gene delivery system. I request Dr. Bajaj to deliver his talk on the topic targeting of triangular neuron cancer immune cell cross-talk using engineered web. Over to you, Dr. Bajaj.
आप कंटिन्यू करिए सर.
Okay, good afternoon. Yes. Okay, I think students are from biotechnology, right? PhD students, MSc students. The postdocs or the senior faculty. So I am from the Regional Center for Biotechnology. I don't know, many of you may not know or you may know, right? This is at Faridabad, not only 20 minutes drive from here. Do come and visit us at our labs, right? So you can see the different infrastructure, research facilities to get to the faculty. MSc students can come for the PhD, and there are many project positions, right, where you can apply. And this is my lab at RCB, for Regional Center for Biotechnology, which is a Institute of Department of Biotechnology. So what I have been there from last 15 years, and my group has been working on 3 different areas. And I am trained as a chemist, but now I have moved to the biotechnology. So what we have been doing, we have been engineering different kind of healthcare materials like lipid-based, hydrogen implants, nanomicelle-based, which can be used for different applications, starting from the drug delivery, antimicrobial therapy, and gene therapy. And what we do is that major area among these things has been on the cancer immunochemotherapy, where we will try to target how we can target the different anti-cancer drugs to the tumor microenvironment, and now how these biomaterials or the drug delivery systems can change the lipid metabolism, and how this lipid metabolism changes then change the tumor microenvironment. So the whole now the lab is working on a chemistry aspect as well as the animal studies in depth, along with the molecular mechanism and the immune component. So, as mentioned here, we start our journey from the chemistry, small molecules, which can self-assemble to make the different kind of biomaterials. So, there is a group 3 students who work on hardcore the chemistry part. Then we test these biomaterials in different animal models and 3 cancer models, which we focus is on the breast cancer, colon cancer, and the liver cancer. And then how these biomaterials change the tumor microenvironment and the lipid metabolism. And in collaboration with the clinician from All India Institute of Medical Sciences, we also do some validations in the tumor tissue in the patient sample as well. So all of you know what is cancer. Cancer is the uncontrolled proliferation of the cells, right? And if you see these 3 review articles, first article came in 2000, right? They are told what are the different hallmarks of the cancer. Many of you have read this, and there were earlier only the 6 hallmarks of the cancer. Now you see with the progression in the cancer research field, then there is another review article came in 2011 where they have added the other hallmarks like the immune component is one of the key hallmarks of the cancer. And then in the recent articles 2022 published in Cancer Discovery, there are other hallmarks are added. So now the disease has become we have understood the disease, and it's such a complex disease, and there are multiple factors that play an important role in this. So what the question what we address in our lab is that how we can alter the host immune system to target this complex tumor microenvironment. What are the different delivery approaches, right, that can be used to limit the toxicity of the drugs? How does the different processes, right, because there are different hallmarks, can we target that in together in a single delivery system? And what are the different metabolic targets that can be used? So what my lab has been doing is that we have been using a scaffold called the bile acids, right? These molecules, as we know, are already there in our body. So you can and if you see interestingly, the students who understand the chemistry part, so this is the hydrophobic part of the bile acids. And these are the 3 hydroxyl terminals which make the hydrophilic. So these, you know, right, when we eat any fatty food, right, these bile acids help in the absorption of the fat molecules, right? So because they have the property to self-assemble, right? They can self-assemble because of their hydrophobicity. So we have been derivatizing these molecules to make different kind of biomaterials like nanomycels or the hydrogen implants. So the students who are in the PhD, they will understand that when you send a grant or you send a paper, there are rejections. So the data which I am going to present today, when we submitted this grant, right, it got rejected with this comment that these nanomaterials are very difficult to prepare. That's why the grant is rejected, right? So when I went back to the committee that I have the preliminary data, I can do that, then they again rejected it, saying that you already have done it, right? So this is what you face, but you have to keep on working when you think that this is the right idea. So I am starting with a drug called docetaxel, right? So this drug is given to the breast cancer patient as the first line chemotherapy. So major challenge with this drug is a high toxicity. So I'll give you an example. We got the approval to work on the mice, rat, and rabbits. We gave a drug which is given to the patient. The same drug formulation we got from AIMS and gave to the rabbits. 2 rabbits we gave 4 mg per kg, right? Not a high dose. All the 2 rabbits died on the next day. We thought that okay, maybe some student had done a mistake or something. We used another 2 rabbits and gave again the 4 mg per kg. All the other 2 rabbits also died. So this, I mean to say that this drug is such a high toxicity, but still it is given to the patient because it is only one of the effective drugs, especially for the triple negative breast cancer patient, which don't have any targeted therapy. So because and now when any drug you select it, you have to be very careful. It should not reach the toxic level, and you cannot reduce the concentration that it should not have any therapeutic effect, right? Your drug should be given that way, so it should be above the therapeutic index, below the toxicity index. And there are so many side effects already with the docetaxel drug. So this was the story of my first PhD student who joined in 15 years back. Now he's in our Harvard. So what he designed is that he synthesized a small molecule where this docetaxel was conjugated to the bile acid scaffold. Right? This is the bile acid scaffold. and we attached a phosphatidylcholine group at the C terminal. Now, what we did is that when we take this molecule with another lipid, we self-assemble that, and you can make the engineered nanomicelles. And these nanoparticles are less than 100 nanometer in size, right? You can see the TM and the DLS study. When you give the esterase, the drug will not get released. So interesting is that when we injected these nanoparticles in the mice, you will not see the free drug in the blood, right? Because your drug is conjugated to the bilateral scaffold. Whereas whatever is available in the market, if you give that, that drug is freely available in the blood. It will go to all the tissues and it will kill everything, including cancer cells. That's why when you see the cancer patient, their hair, you will not see their hair, right? The platelet will go down. So here the drug is conjugated, so there is no free drug available for the toxicity. Now, cancer tissues have the high esterases or proteases. They will cleave this drug from here. So you will see the high drug concentration, cleaved drug concentration in the tumor tissue. So when we did the pharmacokinetics and the biodistribution, we see that the nanoparticles are there in the blood, not the free drug. But in the tumor tissues, you have the free drug available, right? So that's how you can reduce the toxicity. So then we did the anti-cancer activity studies. So how you do that? You take the mice, you inject the cancer cells, right? Mice will make the tumor, then you started giving your treatment. So what we did, you have the control, untreated mice, where the cells are injected, but no treatment is given. So how the tumor kinetics is growing like this, right? Then you give the commercially available drug, which is given to the patient. So this is TS, called text to treat. This is a commercial drug. See here, the kinetics, there is a tumor aggression this much, right? Now you give our nanoparticles, the third group of the mice, and you see the tumor aggression. This is much better than untreated as well as the commercial level drug. More important is that now you see here the body weight change, right? When we give our nanoparticles, there is no body weight change in the mice. But when you give the commercially available drug, you see how the body weight has gone down, right? And you don't get a better survival, right? Whereas when you give the nanoparticles, there is a better survival. So then this is the first time in India it happened that we have done the toxicity study in mice, rat, and rabbit, and because of that we got the approval to work in monkeys. So we compared the drug which is given to the patient and the nanoparticles. And if you see here, this is the drug which is given. We take the blood sample from the monkeys before giving the dose. Then we gave the dose of 20 mg per meter square. This is how monkey you can give the dose. And you can see here the parameters are increasing, right? This parameter. So this has some toxicity, right? Whereas if you see when we give our nanoparticles, there is no toxicity here. And we have done other analyses also like pharmacokinetics in detail in these monkeys. So these are the results published long back in this journal. You people can go and read into for the more details. So now in a cancer cells, we have target only with one drug, right? But if you see the treatment design for the cancer patient is always a combination of drugs which is being used, right? And in the cancer, the people who understand little bit immunology, I say there are two kinds of the cells. There are good immune cells, with are bad immune cells, I will call it, right? So good immune cells are the T cells, the activated T cells. So you need to activate the T cells, okay? But there are bad cells, right? Which help the tumor to grow. So that's what it's called immunosuppression, and this is called the T cell immunity. So there is a tug of war. Always immunosuppression cells will dominate and they will allow the tumor to grow, right? So you have to activate this system in such a way that there is the activation of the T cell immunity. So now to understand that, as it is used always the combination of the drugs. So what we did is that we took the two drugs, right? One we took the docetaxel, which I told you earlier, right, which is anti-cancer drug. Then we took the steroid, which is which is anti-inflammatory. We made their different lipids, right, and combined these lipids. So now we have made a nanomicelles, which is less than 100 nanometer size, which has two drugs. One is the docetaxel, one is the dexamethasone, right? So then we test this, and we have an individual nanoparticle also having the single drug. So we did that experiment. Now you see here, these are the untreated. This is treated with one drug, docetaxel, right? And this is dexamethasone, and this is a combination of docetaxel and dexamethasone. So now you can see here, the combination of the drugs is much better than the individual drug, right? Then we went ahead and see whether what I showed that whether you are activating the T cell immunity and you are depleting the bad cells, which are tumor-associated macrophages. You see here, tumor-associated macrophages and MDSC goes down, right? These are the bad cells, right? And then what happened to the T cells? You see the Th1 cells, CD8 cells, they go high, right? These are that's why you are activating the T cell immunity. You can see the IFN gamma is also increased, right? So then we went into the mechanism. So I will not go into the mechanism to complicate the things of the prostaglandin pathway. So this I can leave it. So now this is the journey. Who is the PhD student? Can you stand? So put up your hand. Only one PhD, two. Okay, two PhD students. So MSc students who are going to do the PhD, this is what happens when you send a paper, right? So this is the story of this paper. We sent this paper, it got rejected by the editor only, right? They didn't send for the review. Then I wrote a 10-page rebuttal letter. So this is a very important study. I sent the rebuttal. Then again they rejected. In that rejection, they told me, you do these experiments, then we will send for the review. Understood? Then I again sent the rebuttal. I told you are telling me these experiments to do. I agree, but these are all your control experiments, right? And it will take me 6 months. After that, you will send to the review. I'm wasting too much time. These are control. They will not change my paper at all. So why don't you send this to the review? If the reviewer say, okay, this paper is for revision, then I will address the reviewer comment and I will address your comments. Why I will waste now 6 months and after the reviewer also reject the paper, right? Then again I send a second rebuttal. Then they sent to the 4 reviewers. One chemistry guy, one biomaterial, one immunology, one cancer. Everything covered. And all were Chinese. This is another problem, right? But good thing that all the 4 reviewers and okay, you revise the paper. Then we took 6-8 months to revise the papers. 2 reviewers said yes. 2 reviewers said again revision. Then at the end, 3 reviewers said yes. And 4th reviewer again rejected the paper. He told no, he was not happy. Then I wrote another email to the editor and then it got accepted. Right? So this you understand now, and this journal has impact factor of 14. Right? This grant was also rejected. Right? The paper also, but got accepted. Right? So you have to keep fighting with the system. Right? Everywhere. Right? So now I have told you the inflammation and the proliferation, right? So another important is the angiogenesis, right? There is a formation of the new blood vessels, right? So we use the similar strategy, right? And here what we do, we did an docetaxel drug and we have taken a combrastatin drug, which is the anti-angiogenic drug. We prepared the nanoparticles, right? And we also prepared a chimeric nanoparticle having both the drugs. And you can see here the same tumor tissues untreated, only docetaxel, only combrastatin, and the combination of the 2 drugs, right? And there is a better tumor regression. And we also went ahead and do all the immune profiling studies and see that we can, if we use this combination of the drugs, we can activate the T cell immunity. So, but the ultimate goal, what we had is, so I will not go to the mechanism. So this paper, this is the old slide. So this also got published in the nanoscale. So now the ultimate goal which I have proposed in the grant, right, was to have a nanoparticle system, chimeric nanoparticle system, having the 3 drugs, which can target the proliferation, which can target the inflammation, which can target the angiogenesis. If I have 3 drugs, that will be the right system to do that. So for that, we selected the 3 drugs. One was the gemcitabine. We started with docetaxel, but we could not make that, right? So because the physical chemistry of these molecules, you have to change your systems. So we selected then gemcitabine, we selected a dexamethasone, and we selected a combrastatin. We made these 3 lipids, which are the bile acids, right? And now you can have all different kind of nanoparticles. Means single drug, gemcitabine, dexamethasone, CA4, and you can have combination of 2, right? Gemdex, GemCA4, and DexCA4. And then you have a triple drug nanoparticles, right? All combinations we had. Then we did all the characterization. So I will not go to the characterization. Then we did the animal studies. Let's go step by step. See the first study. I have Gem and Dex. The tumor growth kinetics, untreated, only gemcitabine, only dexamethasone. So combination of 2 drugs is better than gemcitabine and dexamethasone, right? Then we did dexamethasone and CA4, right? The untreated dexamethasone CA4, dexamethasone CA4, right? Again, the combination is better than. Don't make video for this. This is unpublished data, right? Listen, video will not tell you anything, right? CA4 gem, right? This is CA4, this is gemcitabine, and this is combination of CA4 gemcitabine, right? So see the last one, right? Then there you have to compare the three with the combination. You see these are the dual drug combination, and this is the three drug combination, right? So this is much more effective than the dual drug. Then we went ahead and see that okay, this is a very good system which is activating the M1 macrophages, activating the CD8 cells, activating the granzyme B positive CD4 and CD8 cells. Right now, what we have been doing is that we are taking the cancer cells and injecting subcutaneously to the mice, right? And these are the colon cancer model. Now you can say, sir, well, this is not a real colon cancer model, right? You are injecting the cells and subcutaneously. This is not a colon. Do something in the colon, right? So what we did, we made a model where we had taken the cell and injected in the cecum of the mice, right? You inject the cecum of the mice, you get the colon cancer, right? How you will see the colon cancer? You will see the belly size, they will increase. The fluid will accumulate there, right? Now you start the treatment. So here I am doing the treatment only with the triple drug nanoparticle. I had removed the single and double drug. Now here I am fighting with the reviewers. The reviewer want every study to be done single and double also. Impossible to do, right? So many studies. This model itself is so challenging. Like you can't do in 50 mice, right? Now see here, you have untreated mice where you have done the surgery, but you have not injected anything, right? Then you have untreated where you have injected the cancer cells, right? Your abdomen size will increase, right? And then you give the treatment, the size has gone down, right? Mice are becoming normal. Now, you sacrifice the mice, right? Firstly, you calculate the volume. Here, the whole body fluid will fill, right? So, take out the volume and measure that. You see, untreated mice, size has increased, so there is a volume increase. See, these are healthy mice, colon. Untreated means cancer cells were injected here. And after the treatment, you see normal colon, right? Then you calculate the spleen weight because cancer cell will go to the spleen, right? There will be inflammation. Colon length has reduced. Now it has become longer than the treat, right? Now what will happen? Colon cancer also show the metastasis, right? Right? This breast cancer will show to the lung metastasis. Colon cancer will show to the metastasis to the liver. Means cancer cell will migrate from the colon to the liver, right? So for that now we want to see whether these nanoparticles also target the metastasis or not, right? So what you do is that you inject the cancer cells in the spleen, right? What will happen? You have to shorten the model. You can't have cancer cell injected in the colon. It will take a lot of time to migrate. By the time mice will die, so you can't do that. So you have to make a model. So in that model, what we did, we injected the cancer cell in the spleen. Right? What will happen? Cancer cell will migrate from here and go to the liver. So it will be kind of a metastatic model which is commonly used. Right? So again, we gave the treatment. You can't see here clear picture, but you can see here, right? Healthy liver, and this is a metastatic liver. You see the nodes. You can count the nodes untreated and on the treatment node has gone, right? You see here is a spleen where we have injected the cells, right? After treatment is clear, right? So we again have done lot of studies in this model. I'm not going to the deep, but what we have shown is that we can make a chimeric nanoparticle having the three drugs which target the immune cells also, and it also mitigates the liver metastasis as well as the primary tumor in the cecum. Right. So this is the summary of the presentation where we have made firstly the nanoparticle targeting the proliferation and inflammation, nanoparticle targeting the proliferation and the angiogenesis, and the chimeric nanoparticle targeting the proliferation, angiogenesis, and the inflammation. Right. So with that, I'll thank all of you for your attention. And these are the students who are currently, some of them, it was her PhD work, Poonam. Now she has gone for postdoc at Emory University. And these are our collaborators and the funding from DBT, ICMR, and DST. Thank you so much.
Question from the audience.
So how
how these nanoparticles they were exposed? Like, what was the route of administration?
Intravenous routes.
Intravenous. Any particular reason of taking bile here?
Yeah, because we can easily self-sample. It has a chemistry where you can do so many modifications. Easy, right?
Or any effect on stem cells?
Stem cell we have not checked.
Any questions from students?
Please ask any question. Don't hesitate.
But it was nice presentation.
Yes. Yes.
While administering these drugs, did you find any side effects to them?
No, we have quantified the liver parameters. We didn't see that effect. Yes.
Any other question? एस्ट्रेस जब वो कॉन्जुगेट बना सर बाइल एसिड और
Docetaxel, yes.
सर जब वो एस्ट्रेस में अपना ना लिवर जो कैंसर सेल्स होते हैं उसमें एस्ट्रेस एंजाइम्स ज्यादा होते हैं तो वो उसे क्लीव कर देगा
Right.
सर वो फ्रीली ब्लड में घूमेगा जो नैनो पार्टिकल है बट सर जो हमारा ये ड्रग था वो मतलब कैसे
That because tumor tissue have high esterases. So when these nanoparticles go to the tumor tissue, those esterases will cleave the drug in the tumor tissue. So your drug will be available free in the cancer tissue, not in the blood. You want free drug in the right, otherwise this will not act, right?
When it is conjugated, did you check the activity?
Yeah, no, we have done in vitro activity. So in vitro activity, you will see at higher concentration than the individual drug because it's conjugated. Yes, IC50 will be higher.
This is the complexion process of formulation. In fact, but it was good presentation. Just I would like to know that what about the doses? How you selected the doses
Yes.
of triple combination?
Yes. Right. So what we do basically for any animal experiments, we select a drug that we test in 3 different concentrations, and we see that we don't select a dose that it only shows the complete regression, right? We select an intermediate dose so that it is not toxic and it shows some tumor anti-cancer effect, right? So we have a dose also which clearly clear the tumor, but it is so toxic that it kills the mice before even the untreated. So that optimization always do. And other thing what we have done here, because there are 3 drugs, so in the first single drug we have done dosing every alternate day, right? But here when you have 3 drugs, if you do the alternate day, you will not see the effects, right? It is too much of dose you are giving, right? So there we optimize the dose after gap of 2 days, I mean every 3rd day.
You reduce the dose.
You will reduce the dose, yes. You have to do all this optimization.
The purpose was reduction of dose because of toxicity.
Yes, right. Not because and one is toxicity, and the other is you have to see when do that. If I do a very high concentration with dual drug and triple drug, you will not see the effect, right? You are giving such a high dose, right?
So have you ever seen the synergistic effects of those things?
Synergy in animal model is very, very difficult because you need lot of animals to do the calculation. There is a paper how to calculate. So we have not actually calculated the synergy effect, but in the dual drug,
In vitro studies also.
in vitro there is a problem. So that's what when we submitted this paper to a very high impact journal, so this question came about the synergy. Now here my argument was that it is impossible to get a synergy in vitro system because here my target is not only the cancer cell, right? If I'm talking cancer cell, then I can get the synergy easily, right? But here I'm telling that my nanoparticle targeting cancer cell, immune cell, and the endothelial cells, right? So I have to make that organoid system and then do that. Why I will do that when I'm doing all the study in the animals? So this was another comment. So there was 4 reviewers, 3 said yes, and 1 person has put all the comments.
So whenever we are working, just I'm telling you some example of natural products.
Yes.
So when we are working like in combination, just one study of one company, mainly I'm consulting to many companies also. So once they thought of developing a drug, so at that time we came out with pharmacopoeial commission, and pharmacopoeial commission was recommending to not use the drug in less concentration. So suppose that your tablet is composed of 5 components, you have to use each and every one in the therapeutic window.
Agree.
Okay.
Got it. Yes.
Then when we combined all those things in 5,
Right.
We tested it on 40 rats. None of them we got in 1 hour. All 40 rats done. We reduced the concentration up to 20. 20th, then we got some good result. And mortality was
Yes, yes, that
so
is
like
right.
that. So this is even just one example of black pepper, piperine.
Right, got it. Yes.
And in with Ravalfia, approximately 10 to 15 years back, one of my students was working on Ravalfia. So we are finding some neurological behavioral study was there. So same dose, the reported dose of reserpine that we have given with just 5 milligram per kg of black pepper, paprine. And then animals, 11 rats were died at once. And the same dose, then what we did, that before dying we take out the blood. So when we checked that pharmacokinetic parameters, bioavailability was increased 16 times. So these are the some
Yeah. You
which
told
are-
they are practical challenges. Yes,
yeah,
yes,
so we are working on synergy in fact.
yes, yes, right.
And since plants are composed of multiple components,
Yes, more difficult, very tough,
yeah,
very tough.
very
Yeah.
tough to work
Yeah,
on
yeah.
them.
And very tough.
And pharmacokinetics and pattern recognition, many other things, many challenges. But
Yes,
still, we have a good one, and you have good facility there.
Yes, yes, right.
So many of the students can explore.
Any more
Thank you very
questions?
much.
Any more questions?
So now I invite Professor Saeed Ahmed to hand over a token of appreciation to our speaker, Dr. Avinash.
आ जाइए संजय।
Sir, please.
स्लाइड वाला लगा
इधर ही है।
बैकग्राउंड
ये the background.
आ जाइए सर।
वंडरफुल टॉक
We have one for the presentation from management.
I know where I know.
That's really very right.
No, I don't receive it.
earlier where US or where?
No, I did my MSc from Punjab University in Legal. PhD from IISc Bangalore. Then I did
Very
postdoc.
prestigious. Yeah, I understand. In MIT we have about 13 Ramanujan Fellow and
So now I request Professor Saeed Ahmed to hand over a token of appreciation to Professor Tanveer.
my pleasure.
We two did PhD together.
Now I request Professor Tanveer to
Yeah.
hand over a token of appreciation
Again,
to
it's my
Professor
pleasure.
Saeed.
Now I kindly request our esteemed chairpersons to facilitate Dr. Mohan sir for moderating this session.
Hi, Dr. Mohan. I understand
Group photo.
Yes, yes. Dr. Avinash.
Dr. Monica and other.
You can come and join here.
She should also come.
We'll exchange.
Right.
Thank you so much for your time. Shall we go to Hollywood?
Right? Back. बाद में आ जाएगा।
अरे
बाद में आराम से
वगैरह का क्या
हमारे पास तो चीजें इतनी हो जाती है कि मैं तो छोड़ ही देता हूँ, कहीं से नहीं जाता।
हाँ जी, और अनाउंस all are requested to please move to hall number 1 convention center for the tea.
मैम सेंड कर दूँ।
और कैमरा उठा लेना।
एक सेकंड यार, ये सब
मैम मोमेंटोस डालना।
मोमेंटो भी टेक्निकल होते हो सब वो देंगे थिंकिंग के फेलियर। प्लीज आप लोग वहाँ चले।
यस मैम। ओके आशीष तुम्हारा है ना?
मेरा तो पता नहीं किसने उठा लिया। वो फिर टॉप के पास है ना, एक ले लो आप, उस पे कर लेना आप यार।
मोमेंटो यहाँ रखे हुए हैं।
नहीं चलो लेके तो चलना है। मैं स्पीकर के साथ जा रही हूँ।
ओके
तुम लोग सब
मैं यहाँ
ठीक है। और ये बॉटल्स यार जो बॉटल्स
श्योर निजाम भाई इसको बंद कर दूँ, सेशन एंड कर दूँ, हो गया है।
Yeah.